Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science podcast. I'm Vera, editor-in-chief at Xtalks.com, and this week I'm delighted to introduce a very special guest on the show. We have with us today Dr. Tim Culp, who is the Vice President of Research and Development at Interius BioTherapeutics, which is an early-stage biotech company expanding the potential of cell and gene therapy. And they do this by developing next-generation delivery technology based on the inventions from the lab of Dr. Sar Gill at the University of Pennsylvania. Dr. Culp, thank you so much for joining us today.
Oh, thank you, Vera. I appreciate the opportunity.
So to start us off, can you tell us a little bit about your background in the industry and what led you to join Interius BioTherapeutics as VP of their R&D department?
Sure. It was, as I look back, it wasn't something that was expected, this opportunity. So I'm glad to be here. So I was trained as a virologist, and as a virologist, I was really interested in finding ways that viruses could be used to do good things instead of just make us sick. So during my graduate and postdoctoral work, I became very interested in how viruses could be used to benefit humans instead of just make us sick. So as I was thinking about that, and then I coupled that a little bit with, like you, and I'm sure most of the audience, we've all lost people to cancer. And so there was a real interest and still is an interest in how do we help the immune system? How do we use viruses to help the immune system identify and destroy cancer? And so this was, early 2000s when, CAR T was just beginning to be invented by different groups around the world, including the University of Pennsylvania was instrumental in that as well. So I ended up taking a job at Merck in vaccine development, and I also supported oncolytic virus development. There I worked in both early and late stage programs. But really what I was learning there, in addition to developing a given product, was about the different aspects of product development. What does it take to go from a good idea to an actual product that could be commercialized and help people and be robustly manufactured? There'd be a supply chain that would be reliable. So what did it take to do that? What different functions needed to be involved in that? And it's a huge undertaking, to be sure. So learned that process or learned about that process and the different aspects of that. And then in 2021, I was contacted by a person who had mentored me in my previous job, who had I'd kind of lost touch with a little bit, and I was contacted, and he was telling me about this opportunity at Interius, and I initially wasn't very interested because I had a lot of satisfaction where I was. I had a lot of purpose in what I was doing, but as I thought about it more and talked with my wife, we felt like, let's at least take a step and talk with Phil Johnson, who is the CEO of Interius. So I met with Phil, and he shared a little bit about what the vision was. And as you mentioned, it was the company is a spin out of Sar Gill's lab at the University of Pennsylvania, and the driver, I guess, for it at that time was to meet the need of patients who are currently not getting these ex vivo CAR T cell therapies because of manufacturing issues or because they weren't healthy enough to get through a lymphodepletion prior to treatment. So there's just a lot of variety of things that would lead these patients to not get the help that physicians would want to give them. And so he shared a little bit about that vision and about the possibility of using these viral vectors to educate the immune system to find these tumors and to do that in a way that was more accessible to a wider number of patients, and that would take it from something that's where we're creating a personal therapy for somebody to a therapy that's an off-the-shelf therapy that would just be a huge step forward. And so it was quite an undertaking, but it seemed like the more I talked with Dr. Johnson and looked at the literature, the science had evolved over the last couple of decades in such a way that it was now possible to think that we could put a lentiviral vector into somebody's bloodstream and specifically target genes to a given cell type. And so made the decision to make the leap, and I retired officially from my previous position and came to Interius to oversee these things, and just have been really privileged to be part of an extremely talented, committed, creative, hardworking team that we've done a lot in the last 3 years. I guess it's been since I've been there. We've seen just a huge amount of progress in that as the company's grown, from a small little lab space with a couple of guys to around 50 now, and with a lot of different functions that are involved in drug product development. So I'm glad to be there.
Yeah, just curious, now that you mentioned how the company is growing, where is Interius located?
So we're located in the Pennovation Center. So it's just it's near CHOP, University of Pennsylvania. We have some space there that we leased from the University of Pennsylvania. It's been great space for us. It's very accessible, and we have more space than we could imagine probably for a company our size, which is a great problem to have. So
that's great. And you touched upon this a little bit when you were talking about what motivated you to join Interius, but just in the broader oncology space in general, what are the unmet medical needs in oncology indications, particularly with respect to the application of cell and gene therapies?
Yeah, it's a good question. There are many unmet medical needs. First thing is I want to, so we've got some licensed products, about, I guess, 6 CAR T cell products and many that are being worked on. But there's still an unmet medical need in terms of blood cancers, which is what the current products are serving that population of patients. The reason I say there's still an unmet medical need, even though we have multiple products that have been licensed, is that these are being manufactured on a per patient basis. So every batch that has to be manufactured for a given individual is specific to that individual. It involves their cells being engineered outside of their body, expanded, and then put back into that patient after they've experienced some lymphodepletion prior to putting those back in. So they're patient-specific batches. And so largely because of that manufacturing challenge, I guess, to manufacturing these products, a special batch per patient, only about 1 in 4 patients actually who is eligible for these products actually is able to receive them. So what we're trying to do is there's still some unmet medical need to having to do with blood cancers like multiple myeloma and B-cell lymphoma, et cetera, that we feel like with manufacturing, so to speak, these products in vivo, by putting the lentiviral vector inside the bloodstream, having it be able to target specifically to the cells we want to reprogram to see the cancer, we believe that will hit that unmet medical need or can help with that. We also, we certainly look at solid tumors, and there is a huge need there as well for improved therapies. And there are many companies thinking and working in this space. The issues there is that solid tumors tend to have a tumor environment that is a little bit hostile, I guess, to the immune response. So these cell types have a hard time either infiltrating the tumor, or they have a hard time surviving or remaining active in that space. So there are some new strategies that are being worked on by us and other companies to try to maintain the ability of the immune system to not only target the right cells. If you have a tumor type that has a tumor-specific antigen and you can target it with the immune system through a CAR or something like that, how do we keep those cells active once they get into that environment where the tumor's trying to suppress their their response, and how do you keep those CARs active? So there are different strategies, and in many cases it's some kind of immunomodulation, it would be called, where the immune system is releasing cytokines or something in order to try to keep those cells active. So whether you put those as part of your gene that you reprogram the cells with, or you supply those exogenously, there's a couple of approaches that might work there, but I think there'll be some movement into that space as well, into other solid tumors at least. And that includes brain tumors, because T cells can they can bypass or get through the blood-brain barrier. We also see non-oncology indications that our platform can be helpful in. We're starting with a CAR T payload, so to speak, or delivering that gene to T cells and NK cells. But once we demonstrate that can be done in a IV delivered way, there's other cell types that we've shown at least proof of concept that we can hit specifically, and so you can change out the payload. It's a bit like I'll liken it to a shipping situation where you have a box and you put an address on it, and it says, where do I want this box to go? The box represents the cargo or the gene. And the question is, how do I get that box to the right address? Well, there's a label on it that says this is where you go. And then a shipping company's taking it to that address, dropping it off. That box gets open, and now that cargo is at the right address. Similarly, that's what we're done. These lentiviral vectors that we've designed, this platform, we can put an address on it and say, this is what cell types you're looking for. Go find these and deliver the cargo, the gene, to that particular cell type.
That's great. I was just wondering, can you share any more insights about the proprietary lentiviral vector gene therapy platform? Anything else?
Sure. Yeah. We are always happy to talk about it. So our platform has essentially three key components. One would be what I'll call the binder. That's our typical term for it. This binder is like the address on the label. So it's a molecule that we put on the surface of our lentiviral vector particles that is a high affinity binder that helps that vector particle find the right cell in the body and bind to it. Then the second component of our vector is a fusion protein. A fusogen is what we typically call it. That's been it's been derived from a viral protein that you engineer so that it doesn't bind its native receptor, but it can still fuse one membrane to another, which is how viruses kind of inject, so to speak, their gene into your cell, is they have to do a fusion of some kind, or envelope viruses do, in order to deliver that gene. So what this is effectively is a way to open the door of the right house, if I get back to the imagery. So the binder helps it find the right address. This fusogen helps it open the door. And then the third component is the transgene. What is that gene that I want to deliver? And that gene, in this case, for our first couple of products at least, will be a CAR, a chimeric antigen receptor gene. But you could switch out that. The platform allows you, it's literally a plug and play kind of thing where you can just change one component at a time and mix and match and say, what cells do I want to hit, and what gene do I want to deliver to those cells?
So Dr. Culp, can you tell us in more detail, how does in vivo gene therapy delivery work, and what are its advantages over ex vivo approaches?
Yeah, absolutely. It's so foundational, to where I think the technology is going, and where we're trying to go as a company. So ex vivo CAR or ex vivo cell therapies, I could expand that a little bit beyond CAR, but basically the idea is that you're gonna find the cells that you want to engineer. You're going to remove them from the body, and then outside of the body in this ex vivo environment that you try to make as hospitable as you can to these cells. You're going to put some kind of a vector that will deliver a gene to those, and then later you'll return those to the body, typically after a lymphodepletion of the patient. So what we're trying to do now is to eliminate that whole need to remove those cells outside the body and to just take that same vector or a similar vector that's been more engineered than that initial one and put it in the first product, the bloodstream. You could put it in other compartments of the body, but let's just talk about the bloodstream because that's very accessible. It doesn't take any special equipment. It's something that doesn't take a special trained medical staff, but to deliver something IV into the patient, and then let the vector, which is now targeted specifically to a certain cell type, to find the right cells. So I often will think back about, if you go back 20 years or so when ex vivo CAR T was really kind of getting its feet under it. The founders of these kinds of therapies, many of them at Penn, it would never have been viewed as, oh, this would be the ideal way to do it, is we'll go into the body, we'll take the cells out of the body, we'll engineer them, and then we'll put them back in and have to immunocompromise this person in the meantime. It's kind of the hard way to do it, for lack of a better word. It's difficult on the patient. It's hard to manufacture them. Every product is unique to that patient. It's expensive. It's just not the way you would want it to be if you didn't have to be that way. But the reality is the technology, our understanding of how to get vector delivered, how to get a given gene delivered to the right cell was at a point where you couldn't just put the vector inside the patient because it would go all over the place and have a lot of what we call off-target, gene delivery. So until kind of that technology to how do I target these vectors, until that caught up with the incredible advances that have been made in CAR T cell therapies, which have revolutionized in many regards how we think of treating these patients and what might be possible in the future, until it caught up and is now we're on the threshold. So we're on the threshold of that point where with Interius and other companies have found different strategies that we can do that in vivo and now eliminate that whole piece. So the quick way to think of it, or at least the way we like to think of it at Interius, is faster, better, cheaper. If I had to have a slogan, faster, better, cheaper. So the faster part is this would be an off-the-shelf product. So literally, if you have a facility, a hospital facility that has a freezer that could store a COVID vaccine, that's what they need. Okay, so you're going to take a vial off that. It would be immediately accessible to a patient who qualified for the therapy. The second part of that, or the related part of that, is the accessibility. Democratization is the lovely term for it, but basically, we're going to make this accessible not only to somebody who lives near a world-class hospital like Penn Medicine, but also we're going to make this accessible to somebody who lives in a place that maybe doesn't have that kind of hospital accessible to them or that kind of healthcare. And they don't have to now travel to a tertiary healthcare system to get treated. So that's part of that faster part as well, I guess. The second piece of that is better, and there's a lot of components to that, but I'll emphasize, I guess, the fact that we think we can engineer these cells where they live, so to speak, in the body. These cells, they don't live outside the body. Obviously, they live inside the body. They might live in the bloodstream. They might live in the spleen. They might live in the bone marrow. And by engineering them inside the body, we believe they can be of better quality and higher potency and be more likely to be able to remain as long as those cancer cells remain. And so there's been some recent work in ex vivo CAR cell therapy that has shown that if you can shorten the manufacturing time outside the body, meaning instead of taking something that might take a week and a half or two to expand those cells before you put them back, if you can do that in just a couple of days, those cells perform better. They're better quality, and you don't need as many of them to get that cancer, to destroy the cancer in that person. So the way we look at it is, well, if shorter is better, then the very best way to do this would be to engineer these cells inside the body where they live normally, not to stress them by pulling them out and have to try to ramp them up with a medium that they're not used to growing in, and a conditions that try to be as kind as we can to them. But these cells do really well inside the body. Let's leave them there. Let's engineer them inside the body. And now we have the technology to do that. So that's part of the better. But the other part of the better is at the patient level is with ex vivo CAR T cell therapies. You have to lymphodeplete the individual. You have to, before you introduce that ex vivo CAR cell therapy product back in, you have to knock their immune system down, which is we've heard many testimonies where that's the worst part of it is having to go through yet another round of chemotherapy to knock their immune system down before they return these cells that were expanded outside the body back into the body. And it's been shown, at least for ex vivo, that it's necessary. Our animal studies have shown it's not necessary if you can engineer those cells inside the body and transduce them there with the viral vector. So we believe that is not only possible but supported by the data, and we hope to show that in the clinic, starting later this year. And then the cheaper part is probably obvious, but I'll say it. Is if I don't have to manufacture cells, a cell product specific to a given person, but I can make a big batch that could serve hundreds of people, that's obviously going to be cheaper. And secondly, the hospital costs are going to be much less because this person now doesn't have to stay in the hospital while they're lymphodepleted, waiting for their treatment. They get treated, then they have to remain as their immune system tries to bounce back. So there's all sorts of expenses there as well. So both from a cost of goods, for the product itself, you're making a viral vector instead of the cells. But also from a medical expense perspective, it will be cheaper. So faster, better, cheaper is kind of the way we look at this, the potential here.
Yeah, I thought that was a great overview of the comparison of in vivo versus ex vivo gene therapy approaches. To ask you kind of a broader question, how have you seen the field of gene therapy evolve over the past decade? And in your opinion, what are some upcoming industry trends in gene therapies?
Yeah, so it's a very exciting time, to be sure. So it's a very good question. The most obvious thing, at least that jumps out at me, is just the amount of approvals for licensures for these products is just really ramping up. I think there's almost 40 now in the United States that are licensed, and there's around the world. So we're seeing, I guess I would say it as a kind of a pathway that's being worn. If you imagine in a park and where people walk, you get a pathway, and we've kind of walked that path enough. Companies are walking that path enough, and how to get a gene therapy not only from a good idea to actual licensure has been worn, and so to speak, and I think we're getting better at following that path in terms. And there may be shortcuts, by the way, and there may be better ways to walk it. But so that's one thing that I've noticed. And then the second thing, which we've talked about, which is why Interius has been focused on, as well as others therapies. But there's many companies working on these targeted approaches, so that we can develop either viral vectors or lipid nanoparticles or some kind of particles that can specifically target given cells inside the body. Francis Collins recently was quoted as saying that this was the top priority for gene therapy would be precision target delivery of gene medicine is the idea. And so I think that's the huge, I don't want to call it the holy grail, but it's the goal right now for many companies, where they know what gene they want to deliver, but they can't get that gene there. And so I think you're just seeing an explosion, so to speak, in effort in that space, and thankfully progress as well. And we're glad to be part of that at Interius.
And could you tell us about any recent company news and developments from Interius?
Oh, absolutely. So one of the things we're most excited about right now is not only the progress we've made over the last 3 years, but we now have approvals in Australia and 2 sites that are enrolling patients for our first study. So we we'll have patients dosed by the end of the year, and we're excited to see the performance of the product in humans. Like I said, we've done a lot of animal studies and cured a lot of mice and monkeys, but we're here to help an unmet medical need in humans. So we're excited about that. So you can be looking forward to that news that's going to be hopefully coming out before the end of the year. We begin to see some progress in that study.
Great. And in your opinion, what's next for Interius? Like maybe the next 5 to 10 years?
Yeah. It's a very interesting question and a complex one. So, I try to imagine and have my team imagine what will happen if we demonstrate in humans, and I'll be optimistic after we demonstrate in humans that we can deliver these genes successfully to specific cells without any kind of concerning safety signals. What are the possibilities? What might happen? And it's pretty exciting to think about because not only, I would expect Interius to continue developing additional CAR-based products. Like, for instance, we have our second product will be targeting autoimmune disease using a pathway that's currently being worn again. I'll use that imagery by these ex vivo CAR 19 cell therapies. And bispecifics, and a lot of people in that space right now. But again, if there's something that can be done with ex vivo CAR T, that seems like a very obvious place for us to move. So our second product will be a CAR 19 cargo, a gene for that we hope to have in the clinic by the end of next year, by the end of 2025. Additionally, as I said before, we've demonstrated an ability to hit different cell types, not just T cells and NK cells, but other cell types, and deliver all sorts of genes that, everything from, just a reporter construct to do a proof of concept. But we've also demonstrated that we can do, gene editing. So you can correct a gene, you can deliver a defective gene. So it's not just a CAR-centric opportunity that we see in front of us. We see an opportunity to impact any disease where a correct gene or a correct functioning gene needs to be delivered to a given cell type, or we need to correct a gene that's not functioning correctly, or give a cell a new capability, like we do with CAR Ts, which is we're giving them a gene to tether them to the cancer cells. So anytime you want to deliver a gene to a specific cell type, that's space that we want to be in. So I would say there will be some non-CAR based therapies that we hope to be working in.
And from your perspective, and if you were to combine all your experience, is there any advice you would like to share for aspiring biotech innovators, perhaps for those who are in the gene therapy space or in general?
Yeah, interesting question. So, so as an individual, I would always say be open, and don't think that your prior experience is wasted or is not transferable in some regards. So look for those opportunities because we bring different things and different experiences. In my own case, I was a high school teacher for 15 years, and I coached and all of these things. And on a weekly basis, I see kind of some of those things that I learned during that period are useful. I'll say it that way. In what I do as a scientist and leading R&D at Interius. As far as another thing, it's more for a company, but it has to be applied at the individual level, and that is keep an eye on I'll call it the long view of the company or where you're trying to go. Sometimes we get so excited about the biotechnology, whatever that is, whether it's oftentimes extremely interesting science, I'll say it that way, and it can get to a point where we forget that for this ever to go anywhere, for this ever to actually help a human being somewhere, I've got to think of the long view. How do I get from here to there? And it's a long road, and it's a hard one. It's a challenging one that involves things that are maybe less obvious to think about, like, can I manufacture this? So one of the things we've done at Interius, and I think this is one of the strongest things, that has allowed us to move so quickly to the clinic, is we've invested in building internal process development, drug product development, analytical team internally, which has allowed us to, I believe, have an accelerated pathway, because we've developed a custom process that's ideal for making our product, formulating our product, et cetera, because we know that at some point we're going to want to take this to a regulatory agency and not just say, we want to do a clinical study, but we're going to take it to regulatory agencies and say, we want to license this and have an approved product that can be sold in your jurisdiction, whether that be the EU under the EMA or the FDA over the United States, et cetera. So keep that long view in mind. Don't forget about it, because there's a lot of challenges there that take time to overcome, and sometimes we get too excited or too distracted maybe by the incredible possibility of what we're doing, what we're trying to make, that we forget we also have to make it in a robust way that's reliable. And so I think that's one of the things that I've seen Interius do well, and I think other companies are beginning to think about it more, because they're seeing that many of the things they want to do, they can't, because of manufacturing issues, for instance. So have those thoughts early, because it takes time to develop those in addition to your product.
Well, thank you very much, Dr. Culp, for sharing those insights with us today.
Sure. It's been a real pleasure to talk with you, Vera.
Thank you. That's the end of this week's episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you all for joining us, and we'll see you next week. Bye.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.